Hello, welcome to Vitus Academy. Today we'll be talking about CD8 T-cells and CD4 T-cells. T-cells, what are T-cells? T-cells or T lymphocyte cells are white blood cells that are sourced from the bone marrow but mature in the thymus gland. They are usually distinguished from B cells and natural killer cells due to the T cell receptor on their surfaces and play a central role in cell-mediated immunity. Majority of human T cells rearrange their alpha beta T cells receptors and are termed alpha beta T cells. They are part of the adaptive immune system. The CD4 cells respond to antigens presented on molecules called major histocompatibility complex 2, while CD8 T cells respond to antigens presented on major histocompatibility complex 1. Now we are going to talk about the maturation of T cells. In the thymus, T cells known as thymocytes proliferate and differentiate along developmental pathways that generate functionally distinct subpopulation of mature T cells. There, they are shaped into an effective primary T cell repertoire by an extraordinary pair of selection processes. Now this is an illustration of the development of immune cell. In the red bone marrow, we have the hemocytoblasts. These hemocytoblasts are like the kind of a parent cell or stem cells that generate into either B cells, natural killer cells, or T cells. Now, in the bone marrow, before becoming T cells, B cells, natural killer cells, they first generate into the lymphoid stem cells. Now, these lymphoid stem cells either go ahead to become B cells or natural killer cells. The lymphoid stem cells that become B cells or natural killer cells do not migrate from the bone marrow. They remain in the bone marrow and become B cells or natural killer cells. But if they have to become T cells, they will have to migrate from the bone marrow down to the thymus gland. Now, in the thymus glands, they are acted upon by thymic hormones. These thymic hormones act on the lymphoid stem cells and they begin production of T cells, production and differentiation of T cells. Now, in the thymus, after differentiation and production, they have to leave the thymus after maturation and move into peripheral tissues, blood cells, and other organs rather. Now, this is just a general overview. Now, in the next one, we'll be talking mainly about thymus. This is also another diagrammatic illustration of a T cell maturation. In the thymus, T cells, the CD8 cells, or the CD4 T cells, before getting into the thymus, have to go through the bone marrow. This is the bone marrow. Now, in the bone marrow, they have a common lymphoid progenitor. We've seen that before in the previous diagram. And this is a diagram that is specific for T cell. After leaving the bone marrow, the immature precursor T cells migrate from the bone marrow into the thymus gland. Migrate here into the thymus gland, and now there is a very, very important step they have to go through if they must survive. We call it the positive negative selection process. The positive and negative selection process are very, very important in the sense that when they mature, the reaction of the thymocytes on our own self major histocompatibility complex will determine whether they're going to survive or they're going to die off during the negative and positive selection process. So Now, after the selection process, those who survive, the T cells that survive, will then migrate out of the thymus gland into the peripheral tissues. And you should also note that only 2% of these cells, of T cells, actually survive the maturation selection process. 98% of them die off or are eliminated by a process called apoptosis. So we're going to see more of that in the next. This is the positive and the negative selection. Positive selection permits the survival of only those T cells whose TCR, that is T cell receptors, are capable of recognizing self-MHC molecule. Now this is key, like I said earlier on. For us to survive, our cells should be able to recognize our tissues or the cells in our tissues. Now, if they do not recognize them, it means that they might cause problems for us, autoimmune diseases and inflammation and what have you. So, for us to have T cells that are going to survive, going to play a part in our immune system, those T cells should be able to understand and see our own body cells as part of the family, as part of their family. So now, some cells, many cells, 98% of the cells are not going to survive this. So now, it is thus responsible for the creation of a cell MHC restricted repertoire of T cells. Cells that fail the positive selection are eliminated within the thymus by apoptosis. That is, cells that are not capable or competent enough to recognize our self-MHC molecules are supposed to go. They're supposed to die. Now we have the negative selection. This negative selection eliminates T cells that react too strongly with our self-MHC cells. Now they actually recognize. In this group, they actually recognize, but they react too strongly with our self-MHC or self-peptides, proteins, and that means they will also have to go. Yes, because if they are allowed to survive, they will keep reacting until maybe there's an inflammation, autoimmune inflammation, and we go down with immunologic problems and we begin to have health issues. So this is a very, very extremely important factor in the maturation and generation of T cells that will go into the peripheral system. So that is for the positive and the negative selection. And this is a general overview of T cell activation. T cells have to be activated during, when they are playing their part in the immune system, killing off infected cells, producing cytokines and what have you. Now we have the T cell. The T cells are mobilized when they encounter a cell such as the dendritic cell or B cells that has digested an antigen. The dendritic cells are professional antigen presenting cells, or the B cells. Now that has digested an antigen and is displaying antigen fragment bound to its MHC molecule. So what happens is this: the dendritic cell or the B cell encounter maybe an invader, a bacteria, it could be virus, get them, it digest them, and then present part of that bacteria on its MHC molecule. It could be MHC1, it could be MHC2. If the antigens are presented on MHC1, it means that the CD8 cells are going to do the job, they're going to take over. If the antigens are presented on MHC2, it means that the CD4 cells are going to recognize that cell and do its job of either clean up the cell or releasing cytokines. Now, the cytokines are very key. They are proteins, immune proteins that help the T cells mature. They also serve as a bell in the immune system. A bell that when the T cell actually produces it, other cells, other immune cells are drawn to the side. They are called to order and made to understand that there's an invasion that they should take care of. Now, apart from that, also we have the MHC antigen complex activates the T cell receptor and the T cell secrete hormone. This is the cytokine secreting mode, how cytokines are secreted. The MAC and the MAC antigen complex activates the T cell receptor. The T cell sees the MAC on the antigen but on the surface of the dendritic cell or the B cell. And what it does, it binds to the B cell or the dendritic cell. And what happens then? It secretes cytokines. CD4 cells do this. Cytokines that will then draw the attention of other cells, the CD8 cells, the other macrophages, and what have you. So now this is so we also have the cytokines. Like I said earlier, they also help some of the T cells to also grow. Now, infected cells. When we have infected cells, some T cells become helper T cells, secrete cytokines. Okay, we talked about this, which attract fresh macrophages, neutrophils, other lymphocytes, and other cytokines to direct the recruits once they arrive at the scene. Now, some T cells become cytotoxic cells and track down infected cells infected with viruses. And that's the CD8 T-cell. CD4 T-cell. CD4 T-cells mainly secrete cytokines. Where the CD8 T-cells actually are cytotoxic. The kill infected cell. Infected cells. I'm going to write that, sorry. Infected. Sorry, I don't really have a very good handwriting. All the same. So we go to the next one. Now, the cytotoxic CD8 cells. These cells were first discovered before the CD4 helper T cells. They are known as the cytolytic T cells or the killer T cells. They possess CD8 glycoproteins and recognize only antigens bound to the MPC1 molecule. So, activation occurs when naive CD8 cells interact with professional antigen-presenting cells, such as the mature dendritic cells. So what this is saying in a nutshell is that you can call the CD8 cells cytolytic cells, killer cells, or even cytotoxic cells. Okay. So the CD8 cells are very key. CD8 cells only recognizes antigen presented on MHC1 molecule. CD8 cells only actually understand or recognize cells presented on the MHC1 molecule. The next one, okay, CD8 cells can also kill through three different mechanisms. One, secretion of cytokines. cytokines. We've talked about that before. Now there are different cytokines, mainly two cytokines actually. We have the tumor necrosis factor alpha and the interferon gamma. They have the anti-tumor and the antiviral microbial effects. Now, production and release of cytotoxic granules are very, very key too. They are also another mechanism through which CD8 cells kill the infected cells. So they release cytotoxic granules such as the perforin and the granzyme. Perforin actually forms a pore on the membrane of the target cell, while granzyme, this is perforin, granzyme enters the target cell after the pore has been formed, enters the target cells, shutting down production of the target cell and causing an apoptosis. Apoptosis means death of that cell, natural death of the cell, gradual natural death of the target cell, which the cell sheds a lot of its property, disintegrates little by little. Now we also have the Fas and the Fas-L interaction. Fas and the Fas-L interaction causes the cell to primitize and activate the caspase cascade. Also, the end goal is to also have or instigate the apoptosis pathway of the target cell. So after the Fas and the Fas-L interaction occurs, the cell trimitizes and the caspase cascade is activated and apoptosis is achieved. Now this is a diagrammatic expression of the CD8 T-cell. Infected cell. This is the infected cell. This is the infected cell. The infected cell here, the cell is infected with a virus actually. And the virus, of course, virus takes over the genetic machinery of every cell it enters and produces its own peptide. So the viral DNA here is transcribed into viral mRNA, which then is translated to viral peptides. These peptides are then brought to the surface of the cell, of the infected cell. Understand? This infected cell is recognized through the major histocompatibility complex 1. Remember that CD8 cells only recognizes antigens presented on major histocompatibility complex molecules. So this peptide is presented on the major histocompatibility complex molecule 1, class 1 rather. And then the CD8 cells recognizing it binds to the peptide on it and then activates either the Fas-L or the Fas and the Fas-L interaction, or produces its granzymes, or produces cytotoxic proteins that then disintegrates the cell, and the cell dies. Then we have the CD4 helper T cell. They're actually called helper T cell because majorly they don't really kill cells, they only help to kill cells by secreting cytokines. They also help in the activation of cytotoxic T cells by promoting the bactericidal effects of macrophages. CD4 protein is also found on the surface of the cell. And the HIV-1 uses the CD4 glycoprotein to gain entry into the host cell. Now we have the CD4 cell. This is a macrophage. A macrophage is a bacteria that is an immune cell that engulfs invaders like bacteria and parasites. So when a macrophage is activated due to cytokine production, it engulfs an antigen, destroys or disintegrates that antigen by the use of lysozyme, an enzyme that helps in the disintegration of antigens or proteins. Then part of that antigen is then presented on the cell surface of the macrophage for recognition by the CD4 cell. That is how CD4 cell does its own job. So we hope that this is going to be very helpful to you. From us at Fetus Academy, we say thank you.